Hi, my name is Julieanna, and welcome to Science Fun Week. Today I'm going to be doing the food testing demo. The person performing this experiment is a trained scientist and has all the required personal protective equipment necessary. Please do not attempt this at home. So the materials needed for this demonstration are some test tubes, I have an egg here, water, juice, oil, and a couple of reagents like the Benedicts reagent, ethanol, and the Buret reagent, as well as a couple of droppers.
A Buret test is a chemical test used to determine the presence of a peptide bond in a substance. Peptide bonds are found in proteins, and proteins are important because they build and repair muscles and bones and are used to make hormones and enzymes. Proteins can also be used as an energy source in our bodies. In the Buret reaction, the presence of peptide bonds produces a violet color when treated with Buret reagent.
So what I'm going to do is I'm going to take my egg white and I'm going to add 2 ml of the egg white to my test tube. I'm also going to grab 2 milliliters of water just to compare what a structure with protein would look like and what water would look like, because it shouldn't contain any peptide bonds. And next, I'm going to add 2 milliliters of my Buret reagent to each test tube. Watch closely to see any color changes. So I'm going to add it to the water first. And I'm going to add it to my egg white second. So right away, we can see that the solution turned a violet color, and this indicates that the egg whites have peptide bonds in them, while the water stayed blue. So our water didn't have any peptide bonds. The next test we will be doing is the ethanol emulsion test. So this test tests for the presence of lipids. Lipids perform really important functions in our body. They serve as structural components for our cell membrane, and they also store energy. So in this experiment, I'm going to be first adding 2 drops of oil into my test tube. Next, I'm going to be adding 3 milliliters of ethanol into my test tube. After this, I'm going to shake my test tube for around 5 minutes. Next, I'm going to add 3 milliliters of water to my test tube. And if this milky white emulsion occurs, that means that there's lipids present. So if the test tube was clear, that means there wouldn't be lipids. But right when I added water, we got this really milky white emulsion. The last test that we'll be doing is the Benedicts test. And this test tests for the presence of reducing sugars like glucose or lactose. Glucose is found in a lot of food that we eat, and it's one of our main energy compounds. So glucose is present in fruits as well as bread. So the first part of this test, I will be using orange juice, and I will be putting in 1 milliliter of orange juice into my test tube. I will also be comparing my orange juice with water to just show you the difference between the two. And next, I will be adding my Benedicts reagent to each test tube. Next, I will be putting my test tubes into the water bath, and we can wait for the color to develop. Make sure the test tubes are pointed away from you. I've just taken my tubes out of the water bath, and now I can compare the colors. So the water remained the blue color of the Benedict's reagent, but after heating, the orange juice turned a bright yellow color. And this means that our orange juice has way more sugar than the water. And if it had a lot more sugar, this color would actually turn to a bright red. So the Benedict's test can tell you just how much sugar is in your sample. So yellow means that there's an intermediate amount of sugar.
The organic molecules that we just tested for, like the carbohydrates, proteins, and lipids, are essential for a balanced diet. In order to use the energy contained in these molecules, our bodies have to be able to break down everything that it eats, and our digestive system is responsible for breaking down food to absorb its nutrients.
Thanks for watching, and I hope you enjoyed the rest of Science Fun Week.